So, the other way in which you can use prodrugs for is to lower the water solubility. So, some drugs have a pretty bad taste, and so one way to avoid the bad taste is to be able to increase the hydrophobicity or reduce their water solubility. So, what happens is that when you consume the drug, it starts to dissolve in the tongue. So, if you reduce that dissolution, then it is possible to make it less revolting. Okay. So, the other contrasting concept is to actually improve water solubility. So, here what we would do is to add in hydrophilic groups so that you can improve the water solubility. So, the reason why we would do this is that many drugs are actually given intravenously, and so if they are very hydrophobic, then you will have to give them at very small volumes and fairly high concentrations, and also when you inject it intravenously, sometimes it precipitates out. So, in order to avoid all of this, you want to increase the water solubility. Okay. So, the example that we are going to look at is chloramphenicol. So, let me just draw out the structure of chloramphenicol. So, R equals H is chloramphenicol, and what we could do is we could convert this R into a palmitate. So, palmitate is basically a C double bond O with 14 carbons, CH2 14 times and then CH3. So, it is an extremely long hydrophobic chain, and this is used to reduce the water solubility. And if you want to improve the water solubility, then what you would do is you would do C double bond O CH2 3 times COOH, and this actually improves the water solubility. So, here this is actually given for taste. So, when you are giving this orally, you would give this palmitate molecule, and if you are going to give this intravenously, you would give this carboxylic acid, which is going to get hydrolyzed and produce succinic acid, which is naturally present in the body. Also, sometimes when you have molecules that have been given at the injection site can cause some pain, okay, because of poor solubility, and we also mentioned that it can precipitate. And so, for example, in this molecule called as clindamycin, this drug is very painful when it is injected. So, in order to avoid this, what has been done is to convert this to a phosphate ester. So, because it has a phosphate ester, it makes it quite ionic and water soluble, and so when it is injected, it goes into the bloodstream without any precipitation. And so, because of the better solubility, this is much better tolerated when it is injected. Prodrugs have also been used in targeting of drugs. So, here as we have discussed so far, the main objective of using prodrugs or one of the main objectives of using prodrugs is to minimize side effects. So, one way to do it is to actually make modifications which can help with the drug getting activated only in the area of interest. Okay. So, although it may produce toxic molecules as side effects, these are, since they are localized in a particular organ, the side effects are not present in or do not present themselves in other areas. So, the example here is methenamine, which is whose structure is shown here. It is a stable inactive compound when the pH is more than 5, okay? And when the pH becomes more acidic, then the compound degrades and produces formaldehyde. So, if you can see this molecule, it actually has a CH2 attached here, and this will degrade to produce formaldehyde. And formaldehyde is actually quite irritating, and it can be a little bit toxic. So, this molecule is used in urinary tract infections, because urinary tract infections contain mildly acidic conditions, and formaldehyde is very good at it has antibacterial properties. So, the normal pH of blood being slightly alkaline can be used to our benefit. So, therefore, this molecule actually does not undergo any change in the blood, but once it reaches the infected urinary tract, then encounters urine which is acidic, and as a result this molecule degrades to produce formaldehyde. And once the formaldehyde is produced, since it has known antibacterial properties, it can sort of reduce the infections. So, methenamine is an example of a molecule which degrades only at the area of interest, and this is a very useful way for targeting of drugs. There are also examples of prodrugs of sulfonamides which have been used to target intestinal infections. So, here intestinal infections which are associated with bacteria, and these sulfonamides can be used as their succinyl esters. Okay. So, here is the structure of a molecule which is the active drug is shown here, which is a thiazole, and you can convert it to a succinyl ester, and once the succinyl ester is cleaved in the intestine, then the sulfonamide is produced only in the intestine area. Okay. So, because the succinyl moiety is an acidic group, then the prodrug is actually ionized in the intestine, and as a result, for example, in the succinyl moiety would not be ionized in the stomach because of the pH. So, once it gets into the intestine, because it is ionized in the intestine, it does not get into the bloodstream, and then slow enzymatic hydrolysis of the succinyl group releases the active drug and succinate, which is tolerated very well inside the body. The targeting of drugs is also very useful in tumors. So for example, we have already looked at a lot of antibody drug conjugates, and these antibody drug conjugates can also be viewed as prodrugs in some form. Okay. So we have already discussed this previously, so we will not spend much time, but these antibodies are highly specific towards their antigens, and the tumor cells present these antigens on their cell surface. So the antibody goes and binds to this antigen, and along with it, it can take the drug with it. Okay. So using this concept, there is also another whole field of prodrug therapy which has been developed, which is called antibody directed enzyme prodrug therapy. So there are two important steps in this antibody directed enzyme prodrug therapy, or ADEPT. The first one is what we would need to do is we make an antibody enzyme complex. So here is the antibody and here is the enzyme. Okay. So once you make this antibody enzyme complex, then it is administered to the patient. This antibody is raised such that it is going to go and bind selectively to tumor selective antigens. Okay. Now what we do is that this enzyme is actually a special kind of enzyme which is present only in bacteria. So for example, it is a bacterial carboxypeptidase. Now what happens is that after some time given, then the antibody slowly accumulates in the tumor site. So here is the antibody and here is the bacterial enzyme. Okay. Now once you have this whole complex going and binding to the tumor, then the rest of it which is not binding will be cleared from the blood supply. So you have a situation where this antibody goes and binds to the tumor site. Appended to the antibody is an enzyme. Okay, and this enzyme is not present in humans, it is a bacterial enzyme. Okay. Now if you can send a prodrug to the site of interest, then what happens is that this is the tumor, right, this is the antibody and this is the enzyme. So since this enzyme is present, and enzyme is as we have defined it, is a catalyst, so the prodrug can be activated in situ to produce the actual drug, right. And since the enzyme is actually a bacterial enzyme, this prodrug can be designed such that it is getting cleaved by a bacterial enzyme to enhance the success of this strategy. So therefore the cancer drug which is toxic is produced only in the proximity of the tumor, and so therefore what we can do is we can actually administer the drug at a very high dose and not worry too much about the toxicity. So the example that we can look at is this CJS 149, which is basically a molecule which is activated by bacterial carboxypeptidase. So the carboxypeptidase actually cleaves at this site and produces this amino acid and carbon dioxide. Okay. And here is the active drug, and in this case it is a DNA alkylating agent. So what happens is that if you use this approach that we have shown, so you have an antibody and you have the enzyme, and so this enzyme is going to come and cleave, and this happens only in the presence of a tumor. So, the advantage of this methodology is that it can generate a large number of active drug molecules at the tumor site, and these of course can diffuse and interact with the tumor and hopefully kill it, right. And so therefore, the amount of active drug that is present in the bloodstream goes down, and the amount of active drug that is going to get to distribute across the body is also going to go down. Of course, a lot of research has been carried out on ADEPT, and a number of bacterial enzymes have been identified to carry out this process. So, the example that we looked at is carboxypeptidase, but you can also have penicillin G acylase and beta lactamase. So, as I mentioned earlier, the advantage of using an enzyme that is not present in humans, or which is also called a foreign enzyme, is that this can be the metabolism of the prodrug happens only where this enzyme is present. Okay. So, since it is not present in mammalian cells or normal human cells, there is very little chance that the prodrug will get activated inside a normal cell. Okay. So, what we are doing in this process is we are highly localizing the drug to the tumor. Okay. It is also possible to use foreign enzymes which have counterparts in the body, as long as we are able to sort of figure out the enzyme which is present in extremely low levels in the blood, or which are very structurally distinct. So, for example, we can use enzymes such as beta glucuronidase, which is present in our cells, but it is present at very low concentrations. Or we can also use this enzyme called as nitroreductase, which actually reduces a nitro group to a hydroxylamine or an amine. So, these enzymes, although there are functional counterparts present in humans, but they are not very efficient, or the reaction is actually reversible. That means that a nitro group can get reduced to the amine, but they get oxidized back, but this bacterial enzyme will convert it completely to the amine. So, the example that we are going to look at now is the beta lactamase complex. So, what you do is that we conjugate a monoclonal antibody to beta lactamase, and this DNA alkylating agent, which is shown here, is actually conjugated in the following manner to produce this inactive molecule. So, on its own, this molecule on the left is inactive. Okay. And now, what you can imagine is that it will go, because of the antibody, it will get directed to the site of the tumor, and on the site of the tumor it will get cleaved by this bacterial enzyme and produce the active alkylating agent. Okay. So, this is a beautiful example of ADEPT that can be used to direct prodrug therapy. Okay. Of course, one of the problems associated with ADEPT is that you can have an immune response. So, since you are injecting an antibody, there is always a possibility of antibody with enzyme complex. This can be looked at as a foreign substance, and of course, the enzyme is a foreign protein. So, because of this, you can actually have an immune response which can clear up this complex completely. So, it is for this reason that it is preferable to use human enzymes with prodrugs that are already approved for anti-cancer use. So, some examples of these human enzymes are alkaline phosphatases. You can have the human analog of carboxypeptidase, which is carboxypeptidase A, and as I mentioned earlier, which is beta glucuronidase. Now, beta glucuronidase is, of course, we need to express it at a higher concentration. So, the advantage of using this human enzyme is that the immune response can go down. But of course, the problem with this approach is that the prodrug activation can also occur in various other areas, because these enzymes are also present in these areas. Especially, for example, if they are present in the blood supply, then they would be activated in the blood as well. Another problem associated with ADEPT is insufficient enzyme activity. So, for example, if you conjugate this antibody with an enzyme, right. So, if the antibody is very good at targeting the tumor site, the enzyme still has to be very active. Okay. So, if the enzyme is not very active, then the problem becomes the prodrug will not get efficiently converted to the drug, right. So, this becomes a problem, and therefore, we would need to vary the enzyme isoform, so that we can have the active isozyme of the enzyme, so that we can improve the conversion. Okay. The example that we are looking at is this irinotecan, which has a conjugate, so that you have the human carboxylesterase enzyme, which is isolated from liver. Okay. So, here the isozyme that is concerned has 26 times more activity compared to the HCE1. Okay. Of course, it is still too considered low to be effective for ADEPT. So, therefore, we would use an alternate strategy or a complementary strategy known as GDEPT, which is basically gene directed enzyme prodrug therapy. We shall look at this very shortly, but what happens is that here you can increase the concentration of the isozyme by transfecting the tumor cells with the gene which is responsible for the synthesis of the concerned protein. We will look at it very shortly, but this is a way in which you can increase the concentration of the enzyme or the isozyme in the area of interest. Another problem or another aspect of ADEPT is the time that is given between the administration of the antibody enzyme complex and the prodrug. So, as we discussed, first you would administer the antibody enzyme complex, and this has to go and bind to the tumor. Okay. Now, you also have to make sure that the unbound complex, that is the one that is freely circulating, is actually removed. Otherwise, if it is not, then as soon as you inject the drug or as soon as you consume the drug, it is going to get activated, and then that defeats the purpose of using this therapy. Now, the longer the time that is given, the more chance that the levels of the antibody complex will drop at the tumor site also. So, because the tumor site is also something that is perfectly capable of metabolizing these complexes. So, you need to optimize the time gap between the administration of the antibody enzyme complex and the drug. One way to address this is to use a 3-stage ADEPT. Okay. So, here the antibody enzyme complex is administered as we would do normally. Then you give enough time so that the complex concentrates at the tumor. Then we would add a second antibody. Okay. So, this antibody actually is raised to target the conjugate, that is the original conjugate that we have used. So, this speeds up the clearance from the blood. So, once this second antibody comes in, then it is going to conjugate to this, and that is cleared from the blood supply. The second antibody can be galactosylated. So, now galactosylated is going to help increase the clearance rate, but it has time to target the circulating conjugate, but it does not survive long enough to penetrate the tumor. Then we add the prodrug. So, here the tumor alone has this or has a considerable amount of this antibody drug enzyme conjugate. Okay. Another interesting strategy to prodrug therapy is antibody directed abzyme prodrug therapy, which is called as ADAPT. So, abzymes are antibodies which have catalytic properties. So, these are relatively new in the literature, maybe 20-25 years old, and there are a number of antibodies which have catalytic properties. So, the concept is to be able to exploit the catalytic property of these abzymes, so that you can activate the prodrug in the proximity of the tumor. So, what we do is that we can design a transition state analog. We have already looked at several examples of transition state analogs. So, we would immunize the mice with a transition state analog of the reaction. Then once you isolate the monoclonal antibodies by hybridization techniques, then you have your thing set up for ADAPT. So, since the antibody targets the prodrug rather than the antigen on the cancer cell line, this is not going to target the cancer cells. However, if you construct a hybrid antibody, okay, so this hybrid antibody where you have one arm, so here this is going to be targeting the tumor and this is going to be the abzyme, right. Therefore, if you convert one of these arms into an abzyme, then what happens is that this is the antibody which has which goes and recognizes the cancer cells, and it also has the catalytic property which is going to activate the prodrug. So, of course, this stage is this approach is still in its very early stages, but it has several advantages over ADEPT. So, for example, it should be possible to design a catalytic mechanism that do not occur naturally. Okay. So, if you have a good understanding of how antibodies work, then you can use this understanding to design new catalytic mechanisms. It also allows for highly selective activation of prodrugs at the tumor site. Now, since the antibodies are raised using human like proteins or human like situations, it reduces the risk of an immune response that is associated with for example, bacterial enzymes. So, at present the catalytic activity of AbZymes is too low for it to be useful. So, there are a number of researchers who are working on improving the catalytic properties of AbZymes so that you can use it for ADAPT. The next major concept of prodrug therapy is gene directed enzyme prodrug therapy. We just looked at it briefly a few slides back, but what this concept is basically what we try to do here is to deliver a gene to the cancer cell. So, once the gene is delivered, then the gene goes into the nucleus and expresses for a particular protein. Okay. So, this protein is the one that is going to convert the prodrug to the active drug. Okay. So, this is the entire concept. Now, since the enzyme will be produced inside the cell, the prodrug will have to get into the cell. So, all of this stuff has to happen within the cell. So, one of the main challenges associated with GDEPT is how do you get this gene into the nucleus. Okay. So, one way to do this is to package the gene inside a virus. Okay. So, as we all know, viruses are excellent vehicles which can go and transfect and incorporate genes into normal cells or into humans. Okay. So, what we would use is we would use a retrovirus or an adenovirus with the gene that of interest that is packaged inside. So, once it gets in, then the desired genes would be spliced into the viral DNA such that the virus inserts into the host cell DNA on infection. Now, the virus is also genetically modified so that it is no longer it is not virulent, that means it does not harm the normal cell. Okay. So, then what it acts as is a delivery vehicle, right. So, of course, there are a number of non-viral vectors that have also been used such as cationic lipids and peptides, but here the problem has always been the selectivity. So, the selectivity of cancer versus normal cells is a huge problem. So, therefore, the delivery vehicle has to be injected somewhere near the tumor site so that you can reduce the normal cells being affected. So, the new enzymes which are ultimately produced inside the cells should not be present in normal cells. Okay. So, because if they are present in normal cells, then your prodrug therapy will not work very well because they will get activated in the normal cell as well. So, therefore, the choice of these new enzymes is very important. Okay. So, one advantage of GDEPT over ADEPT is that since the foreign enzymes are generated inside the cancer cells, they are hidden from the immune response. So, the example we are going to look at is this thymidine kinase, which is produced in herpes simplex virus and has been studied quite extensively for GDEPT. So, the enzyme as shown here is actually going to the viral counterpart of it is going to do a phosphorylation of the prodrug. Okay. So, here the cellular thymidine kinase keeps phosphorylating these groups. So, as these drugs are very poor substrates for mammalian thymidine kinase, the activation will only be significant in tumor cells containing the viral form of the enzyme. So, a number of clinical trials have been presently underway to test this approach. Of course, one problem associated with GDEPT it is that it is unlikely that all the tumor cells will receive the necessary gene to activate the protein. So, let us say you have a tumor such as this which has a number of these cells, right. So, let us say you are able to deliver it only to a few of these cells. So, only a few of them are going to activate or they are going to produce the enzyme, right. So, therefore, there is an important concept that comes here which is called as the bystander effect. So, once the drug is active, as the prodrug is activated inside one of the tumor cells and produces the active drug, then it is somehow transferred between the cells in a tumor. And so, the tumors which do not have the gene transfected are still going to get killed because of the drug that is present in the neighboring cells. So, this is called as the bystander effect. So, this may occur by a variety of means, such as release of the activated drug from the infected cell, direct transfer through intracellular gap junctions, or by the release of drug carrying vesicles following cell death. GDEPT has been used to introduce genes of bacterial enzymes such as nitroreductase and carboxypeptidase G2. So, we have already looked at these two enzymes previously. So, here the nitroreductase enzyme or carboxypeptidase is produced only in the cancer cell, and since the basal level of reduction of these is not going to be very high in the case of nitroreductase, or cleavage of the peptide bond is not going to be very efficient in the case of carboxypeptidase G2, this will produce only the active drug in the tumor cell. One of the problems with carboxypeptidase G2 is the difficulty of having these prodrugs cross the cell membrane. So, in order to overcome this problem, the gene was modified such that the resulting enzyme was incorporated into the cell membrane with the active site, rather than it being able to cross the membrane. So, therefore, the gene was modified such that the resulting enzyme could be incorporated in the cell membrane rather than the inside the cell. One of the examples of GDEPT is this camptothecin derivative. So, here camptothecin is the where you have all of these R groups being hydrogen, and what we could do is we could make this irinotecan, which is basically a prodrug, and here R3 as shown here is actually modified as a carbamate. So, this is actually a prodrug, and once it gets cleaved by carboxypeptidase, it would release the active drug. Now, so we need to introduce the gene encoding the more active carboxypeptidase into tumor cells. So, for example, the rabbit liver carboxypeptidase is about 100 to 1000 times more efficient than the human form of the enzyme. So, therefore, we could deliver the gene encoding the rabbit liver carboxypeptidase, so that it is more efficient than the human form in turning over this substrate. So, there are a number of proton pump inhibitors which are used as in the form of prodrugs, which are activated by acidic conditions. We will look at this later during our case studies. There are examples of prodrugs which will increase the chemical stability. So, for example, ampicillin, which is shown below, is actually decomposes in concentrated aqueous solution because of the intramolecular attack of the side chains. Okay. So, in order to avoid this, what we do is we convert this into a locked nitrogen form. So, here is the prodrug form, and the prodrug which sort of decomposes to give you acetone and the active ampicillin. So, once the prodrug has been administered, it slowly decomposes to release ampicillin and acetone. So, this is one of the ways in which you can increase the chemical stability of the drug. Now, conventional prodrugs are inactive compounds and which are normally metabolized in the body to the active form. A variation of this prodrug approach is to use what is known as a sleeping agent. So, here the sleeping agent is an inactive compound which is converted to the active compound only by some external influence. So, the best example of this is the concept of photodynamic therapy. So, here in photodynamic therapy, there are molecules known as porphyrins or chlorins which are used. So, what we do here is we use this molecules known as porphyrin or chlorins. So, here is an example of a porphyrin like molecule. Here these molecules inside our normal cells are actually bound to metal ions. So, here is a metal ion that is typically present, and once this metal ion is going to be chelated to this porphyrin, these are extremely stable. So, the examples are magnesium in chlorophyll or iron in hemoglobin, and since these are naturally present, they are extremely stable, and in this form they are actually quite well tolerated and they are non-toxic. However, when they do not have the central ion, then these agents become very toxic. Okay. So, when the agents are given intravenously, they accumulate within cells and they have some selectivity towards tumor cells. And of course, these agents themselves have very little effect, but if they are irradiated with red light or a red laser, then the porphyrins are converted to an excited state, and this excited state reacts with molecular oxygen to produce highly toxic singlet oxygen. So, oxygen produces singlet oxygen. Okay. So, once you have the singlet oxygen, then the singlet oxygen is considered as among the most reactive species, and it's going to indiscriminately generate radicals which is going to kill the cell. Okay. So, the singlet oxygen basically reacts with unsaturated lipids or proteins, and also which can further react with DNA, and all of this is going to be extremely toxic. And some of these effects are mediated by hydroxyl radicals. We have already looked at some of these examples previously when we were dealing with chain cutters as a way to target cancers. Okay. So, the example here we are looking at is the steam of porphyrin, and here this is a photosensitizing agent and is used to treat advanced head and neck tumors, and these are given in cases where these tumors do not respond to other forms of treatment. So, unfortunately, the problem with porphyrin structures is that they are inherently hydrophobic and they make them very difficult to formulate. So, what is done is that they use liposomes or oils or polymeric micelles to make these molecules to be water soluble, to be solubilized. Of course, this also has an advantage in using these methodologies, is because these tumors are known to have enhanced permeation and retention effect. So, they engulf and retain these macromolecules more readily than would be the case with normal tissue. And this is because the tumor vasculature is quite random and they are quite leaky as well. So, larger molecules are absorbed better, and therefore, when compared to normal cells, these are much better at permeation as well as retention. So, this is called as the EPR effect, which we will be looking at later. So, despite this, there are some problems associated with this. So, for example, the liposomes which carry this agent can be engulfed and destroyed by cells in the endothelial system. However, the most serious disadvantage with PDT is the photosensitivity. So, once the drug has been released from the liposome and activated, it is free to circulate around the body, and it accumulates in the eyes and the skin. Now, when the person goes out in the sun, then there is going to be activation of these molecules to produce singlet oxygen, and therefore, the patient becomes highly sensitive to light. In fact, this is the property that helped with discovery of porphyrins in photodynamic therapy. So, there is a disease called as porphyria, in which there is a genetic problem wherein the person accumulates porphyrin in skin, okay, and this results in photosensitization and disfigurement. So, the victims of this disease are unable to tolerate sunlight, and disfigurements can include erosion of the gums to reveal red fang-like teeth. So, now you can think about all the vampire stories that you have heard before, where these people do not want to go out in the sun and they want to they come out only in the night and they have red and fang-like teeth. Okay. So, the photosensitivity is a huge problem. So, people are trying to figure out how to improve the method of delivering this agent. Another example of photosensitizer is 5-amino-levulinic acid, whose structure is shown here. So, here this molecule is used to treat skin blemishes which may actually turn cancerous. So, it is a biosynthetic precursor of porphyrins, and once you apply this, what happens is that it starts to synthesize this porphyrins and in the affected area, and then you can do photodynamic therapy with this molecule. Okay, so the next concept that we are going to look at is drug alliances. So, here there are some drugs which affect the activity or the properties, especially the pharmacokinetic properties of other drugs, and so a knowledge of this helps us actually put it to good use. So, what happens is that a second drug is administered along with the principal drug, and this can be done in order to guard it or assist it. So, for example, if there is an enzyme that metabolizes the principal drug and makes it inactive, then you send a second drug along with this first drug, so that it prevents or inhibits the enzyme. Okay. So, the classic example here is clavulanic acid, which inhibits the enzyme beta lactamase. Okay. And this is very useful when we give penicillins as a drug. So, clavulanic acid was isolated from this species by Biochemistry in 1976. It itself had very weak and perhaps not very important antibiotic activity, but it is an extremely powerful and irreversible inhibitor of most beta lactamases. So, here is the structure of the molecule, and you have an enol ether, the sulfur is replaced by oxygen, and you do not have a side chain amino acid. And here is the structure of amoxicillin, which is very susceptible to beta lactamases. So, the strategy that we would employ is to give amoxicillin along with this clavulanic acid. So, what happens is that once beta lactamases are expressed, then this beta lactamases actually cleave this clavulanic acid in preference to amoxicillin, and therefore amoxicillin is protected so that it can display its antibacterial activity. Another example is the antiviral preparation Caletra, okay, which is used in the treatment of AIDS. It is a combination of two drugs, which is ritonavir and lopinavir. So, these two drugs have very good anti-cancer activity, but what happens is that one of these drugs, that is lopinavir, is actually metabolized by cytochrome P450, the isoform is CYP3A4, and in order to protect this, we administer this other molecule along with it. Another example which is in the treatment of Parkinson's disease, we just looked at it, the levodopa example, so which is basically a prodrug of dopamine. However, in order for levodopa to be effective, you need a very high dosage, that is about 3 to 8 grams per day. And over a period of time, these dose levels lead to severe side effects such as nausea and vomiting. So, just to remind you, this is levodopa is converted by a carboxylase, decarboxylase to produce dopamine. Now, levodopa is susceptible to the enzyme dopa decarboxylase, and as a result, much of levodopa that is administered is decarboxylated to dopamine even before it reaches the central nervous system. So, this buildup of dopamine in the peripheral blood supply leads to the observed problems. So, one way is to actually inhibit this dopa decarboxylase in areas which are other than the brain. So, what is done is that this molecule carbidopa is an inhibitor of this dopa decarboxylase. And so, this molecule, when it is given along with the parent drug, allows for smaller dose of levodopa to be used. Furthermore, because it is a highly polar compound which contains two phenolic groups as shown here and a hydrazine, this molecule is unable to cross the blood brain barrier. So, all the inhibition of this dopa decarboxylase to produce dopamine happens outside of the brain. So, carbidopa is marketed as a mixture of levodopa and is also called as co-chiral dopa. There are several important peptides and proteins which can be used as drugs, okay? And the one way to do this is to actually prevent the activity of proteases. So, because all these peptides and proteins are actually going to be cleaved by proteases. So, what we could do is to administer these drugs along with protease enzymes. So, the example here is to use this molecule called as candoxactril, which is actually a protease inhibitor, and it is presently under clinical evaluation. Another example of a prodrug concept is to localize a drug's area of activity. So, adrenaline is an example of a drug which has been used to localize the area of activity of another drug. So, we are talking about interaction between two drugs. So, when injected with the local anesthetic procaine, adrenaline constricts the blood vessels in the vicinity of the injection. So, what happens is that procaine is not distributed across in using the blood supply. So, you would inject adrenaline along with procaine so that you can increase the presence of the anesthetic locally. Another strategy that is used is to help with increased absorption. So, this drug metoclopramide is administered along with analgesics in the treatment of migraine. So, what this drug does is that it increases gastric motility, leading to faster absorption of the analgesic and quicker pain relief.